Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Aldevron Case Study, Validation of mRNA Concentration Determination Using Variable Path Length Spectroscopy. I'm David McCall, Senior Editor at BioInsights, and joining me today are Joe Ferraiolo and Travis Alvine, who will share their strategy and the results obtained from the platform mRNA content assay, specifically focused on mRNA concentration. After the presentation, we'll have a live Q&A session with Joe, Travis, and John Long. And we invite you, our audience, to pose your questions to our speakers using the Ask a Question box at the bottom of your screen. And we'll try to get to all of those during the session. Now I'd like to introduce you to our presenters. Joe Ferraiolo leads the Bioanalytics Applications team and is in charge of the SoloVPE variable path length spectroscopy system for at-line applications. He specializes in UV analysis and leads the development and commercialization of high-value products and flexible solutions that address critical steps in the production of biologics. Travis Alvine leads the Analytical Method Validation Group and manages analytical method validation, method transfer, and method implementation into the quality control release laboratory at Aldevron's Fargo location. He's been with Aldevron for over 6 years, with experience in analytical method development and validation over that time. John Long holds the position of Director Analytical Method at Aldevron and has over 20 years of experience working in analytical methods for large molecule therapeutics, including vaccines, biologics, and gene therapy products. So without further ado, I'll hand over to Joe to kick off today's presentation.
Thank you for the introduction. Hi, everyone. My name is Joe Ferraiolo. I am the Director of Bioanalytics Applications for Repligen. I would like to thank you and welcome you all to the Cell and Gene Therapy Insights webinar with Repligen and Aldevron. My portion of this webinar will introduce the CTech SoloVPE technology and its benefits, how it works compared to traditional UV, and where we can introduce our approach for mRNA concentration determination using our SoloVPE system. We'll show you how you can implement dilution-free analytics to reduce deviations, especially when using technology on the manufacturing floor, and how you can adopt the SoloVPE across multiple sites and groups. At the end of the webinar, we'll have a Q&A session where Travis and I can answer your questions. So Repligen offers some innovative solutions for all the key stages of bioprocessing, from upstream intensification methods using ATF and TFDF through chromatography. Systems focus on improving high yield and well known, obviously, for our OPUS columns. We continue to provide systems that improve productivity in the downstream process filtration and formulation systems that are scalable process development to our productions. Our comprehensive fluid management offerings allow you to be confident with secure fittings and secure supply chains. Our goal at Repligen is to drive integrated smart systems that can be PAT automated using variable path length technology. By integrating our inline flow VPX systems in each of the systems you see here, customers will now have process insights like they never had before and will be able to control their process based on real-time analytics. This is a typical mRNA workflow. The tech used in all the steps for our analytics portion must be purified material, obviously, but we see most people skipping the second chrome step and following up in another change in the past few months that they're combining the CAPI into in vitro transcription, so there's only one chrome during the full RNA production process. The key takeaway here is that wherever there is process monitoring steps, you can introduce our inline analytics to process monitor in real time without any sample manipulation or dilution error. Challenges with traditional UV spectroscopies have been present in the market for decades. The obvious issues with some traditional UV methods includes the assay times for sample prep, where this can take anywhere from 30 minutes to 3 hours, depending on how many samples you're testing. Additionally, samples that need to be sent out for analysis are now bound by the wait times associated by how long and where those groups are on site to be able to perform that work and report the values back. I'll touch a little bit on the manual dilutions versus volumetric in the next slide. But what the real pain point and takeaway here outside of the time spent with the sample prep is the dilution error from analyst to analyst, which can range anywhere from 5 to 20%. Volumetric approaches entirely depend on the skill set of the analyst. As we all know, analyst to analyst variability within the sample prep can be problematic in the results not being consistent from batch to batch or lot to lot. Gravimetrically, certainly more accurate than the volumetric approach, but now we're giving up the time associated with it. So we're losing time. We're relying on the calibrations of the scales being used, the skill sets between operators, and the complex viscosity issues with some of the higher concentration samples that we would be working with in the lab. Our approach for variable path length slope spectroscopy is to take this technique from what was a 7-step operation into a 2-minute, 2-step operation, measuring and reporting. There are no estimations, dilutions, or manual calculations built into our analytics. You get real-time results, easy to validate within GMP environments, and proven linearity within the sample to guarantee the accuracy of the measurement being made. With traditional uv-vis spectroscopy, you're limited by the fixed path length of the cuvette being used and the sample concentration dependent on the linear range of the light source and detector. In this case, if the sample is within the linear range of the path length being used, the result will be an absorbance value, which will then be divided by your extinction coefficient equal concentration. However, if the sample is not within the linear range of the instrument, then serial dilutions must be performed. User prep and handling the issues that I mentioned on the previous slide all come into play, which increases the variability of the results and makes it nearly impossible to platform as a method. Our variable path length spectroscopy method is still a UV-based method. We are implementing 2 changes. 1, the path length is now variable. The light source will come from the Carry 60 spectrophotometer through a fiber optic component called a fibrette. The light source, the path length, is determined by where the light emits with the fibrette to the bottom of the vessel. Using this technique, we're able to utilize the SoloVPE ranging from path lengths of 5 micron to 15 millimeter, and as small as a 5 micron step. This approach allows the concentration to be a fixed variable now within the component. So there's no reason or need to serial dilute your samples, and we rely on the system's ability to dynamically change path length to determine the best linear fit within the sample. The second thing we do differently is we're not interested in taking single absorbance measurements. What we are interested in is collecting multiple data points to plot slope regressions. I'll discuss the technique in the subsequent slides, but all of our measurements will be utilizing a slope regression to calculate concentration or OD values moving forward. The SoloVPE uses the Carry 60 as its light source. It is still a spectrophotometer that can be used for traditional UV methods. However, in our case, we are utilizing the light source via a fiber optic cable to bring the light source into the SoloVPE itself. Once the user presses the Start Collect button, that fiber material called the fibrette will lower to 0 path length. Now, it's important to note this fiber optic is not capillary, it's not hollow. This is a way for us to dynamically change the light path in the sample. Nothing's actually happening to the sample itself. It still remains resident. Once the fibrette zeroes at the bottom of the vessel, The search algorithm will dynamically change from small path lengths to large path lengths, monitoring for one absorbance. It's our target absorbance threshold within the software. Once the software finds the one absorbance path length, it will then collect between 5 to 10 data points worth of collection data to plot the slope regression. It's important to know that this process is completely automated. There is no adjustment of the path length or search algorithm dynamics that have to be entered by the user in the software. The software will dynamically change the path lengths based on how high or low the absorbance values are within the initial search algorithm. Meaning, if the software determines it has a very high absorbance change, it will automatically adjust to very small path lengths, typically in microns. Subsequently, the other way around, if the software determines a very low absorbance contribution from path length to path length, it knows it has a dilute sample and will auto adjust to take very long path length steps. Data acquisition takes less than 1 minute to generate between 5 to 10 data points of regression data. So as you can see in this graph here, the above image shows the slope regression itself, which would be our OD value. This slope regression then will be divided by the extinction coefficient to calculate the concentration. The spectra you see in the bottom graph is just represented each one of these path lengths spectrally. So since we have a scanning spectrophotometer at our capabilities, you're able to dynamically take spectra for the entire wavelength range of 190 nanometers to 1100 nanometers. So to summarize, the automated calculations based on slope, not absolute absorbance, allow us on the right side to see how the data is calculated in the report. We have our path length range, which was used to generate the slope regression, in this case being 28.5. We show the valid R-squared value of 3/9. Slope divided by the extinction coefficient of 1.4 gives us the final concentration of over 200 mg per mil. This can all be done in an automated solution and a validated stored method that gives you the push-button analytics to generate slope-based linear data. We've made tremendous strides within the industry with our technology being used primarily in GMP environments. Additionally, we've been written into the ASTM standard 16916 for general techniques of uv-vis quantitative analysis, which now includes a statement to avoid the dilution step. The instrument may contain an automatic system which will allow the adjustment of path length measurement cell to optimize the measured absorbance. This is slope spectroscopy and variable path length technique being written into a quality standard. So certainly the adoption of our technique throughout the industry is now very well known. To summarize, we're increasing our process understanding in process development stages to accelerate product to market. You are now able to get a true view of your entire process or bench analytics to know exactly where you are within a given process. We've also reduced the cycling time, increased process efficiency and yield by removing all the delays and time spent related to sample prep, investigation of erroneous errors related to dilution and sample prep handling. We've strengthened your process control with high quality and reproducible results as our software is 21 Part 11 compliant. And we're now minimizing the risk with continuous monitoring throughout the entire process. At this time, I'm going to hand it over to my collaborator, Travis Alvine, who is the Senior Manager of Analytical Method Validation with Aldevron.
Thank you, Joe. I appreciate the introduction. I do want to take a minute straight away just to thank Joe and the Repligen folks for allowing this collaboration, allowing me this opportunity to talk about some of the things that we're doing when it comes to method validation in Aldevron, and also just generally highlight kind of where Aldevron sits in the RNA space, and really demonstrate how we're advancing technologies every single day. And so with that, I'll jump into my set of the presentation. I will start with a bit of a high-level overview of what Aldevron is, where we are, where we came from, what we tend to do. Then transition into a bit about method validation in general. I think it's a topic that is worth discussing and really will set the foundation for the last part of the talk, as sort of a case study in practice, how Aldevron approaches method validation and how we continually try to innovate and bring on new technologies that allow us to make these platform methods at the end of the day to support our clients and their process. And so with that, I'll jump straight in. Aldevron was founded in 1998 at the campus of North Dakota State University by Michael Chambers and John Valentine. Aldevron currently is a leading global manufacturer of plasmid DNA, mRNA, gene editing enzymes, and other proteins. We're at a very interesting time in Aldevron's life. We have grown to be the world's largest GMP plasmid DNA facility, located still today, the headquarters in Fargo, North Dakota. We've grown to well over 1,000 employees across all of the Aldevron sites, and Aldevron continues to be rapidly expanding. One of the exciting parts is we've reached a milestone in Aldevron's history, and that we've been in this market for 25 years. We've been in business for 25 years. And so over those 25 years, Aldevron has been a catalyst in advancing therapeutic and vaccine development through expert manufacturing at scales that perhaps our competitors aren't able to do, delivering high quality products, working directly with many of our clients in the scientific community to really support our clients through discovery into commercialization. So I think one of the real interesting things about Aldevron is that in Fargo, North Dakota, we're able to support clients and their progress and their programs through RG manufacturing all the way through commercial to commercial. And so it's quite exciting to see the capabilities that Aldevron is able to give to their clients. And it all happens in Fargo, North Dakota at the GMP scale. Aldevron typically works in sort of three buckets of products from plasmid DNA, protein, and mRNA. And within those general buckets, certainly Aldevron continues to innovate. And so there's been quite a bit of news recently about LNP encapsulation and fill finish. That's something Aldevron's recently brought on at a GMP scale. We certainly support gene and cell therapies by producing CRISPR RNA-based genome editing products. And then our nanoplasmid business located in Lincoln, Nebraska, is also innovating on the plasmid DNA side. What I'm showing here is Aldevron's main campus in Fargo, North Dakota. This is the world's largest GMP plasmid DNA facility in the world. And so really what I'm trying to demonstrate to you here is the size and the footprint that Aldevron has at its headquarters in Fargo. We are a significant player in the CDMO space. We have the capabilities to manufacture at very large scale. And Aldevron continues to grow very quickly. And so when I first started with Aldevron, these buildings didn't exist. And so really this demonstrates not only how fast Aldevron is growing, but also the size and the footprint at which we currently have at Aldevron in Fargo. If we should focus a little bit to mRNA specifically, Aldevron isn't new to mRNA GMP manufacturing. We've been doing this since 2018. We've produced several mRNA batches at GMP scale. We've certainly leveraged our expertise, our historical expertise with plasmid DNA, making that linear template, and then certainly adding on the IVT reaction to produce the mRNA. We have a lot of experience with manufacturing more traditional mRNA and also saRNA, which can be quite complicated. I think when we think about RNA therapeutics and perhaps where the industry is going, we spend a lot of time thinking about the expectation is the RNA therapeutics are going to get much more complex. And so this will mean, larger, longer RNA molecules, adding in additional therapeutic modalities into a single molecule. And really what that means from an Aldevron perspective is is certainly keeping up with the manufacturing and innovate unique and creative ways on the manufacturing side, but it also means the analytical folks and the analytical techniques need to keep pace, right, with the industry. And so we do that typically by working with folks like Joe and his team to bring on these innovative technologies to allow us to make platform analytical methods to support whatever our clients are really working with and whatever our client needs are. If we stay focused a bit on the mRNA workflow, the operational platform at Aldevron, we do offer a complete manufacture of mRNA drug substance and drug product. I won't talk too much today about the drug product and the LNP expansion. There is an upcoming webinar that will cover that. And so I will mainly be talking about, at least from the analytical perspective, everything up until that point. And so what you can see here is that Aldevron is uniquely positioned in that we're able to go from sequence to finished product under one roof. And that's quite advantageous for our clients. It allows us sort of a single source of truth when it comes to working directly with our clients to go all the way from that sequence to the finished drug product. And this makes a lot of sense. I think really Aldevron has been able to leverage what we've been good at for a very long time when it comes to being a GMP manufacturer of plasmid DNA, linear template. Bringing on the capacity to produce IVT mRNA, of course, was critical and something that we've succeeded at. And we've also leveraged our protein business as well and our protein experience, where we're able to utilize in-house manufactured IVT enzymes to integrate into this process. So really, we are controlling this process from start to finish. It allows for great flexibility and adaptability, which is quite important in the mRNA space. Since Aldevron has been purchased by Danaher, it's really allowed us to expand into this drug product space. And so this LNP encapsulation into fill finish, this was really spurred on by a cross-opco collaboration with the Danaher family. And so that really allowed Aldevron to quickly move into that space. One thing that I think of from an analytical perspective is that we're trying to sort of differentiate ourselves. Aldevron is really focused on analytics, and we're really planning for analytics to be a differentiator specifically in the mRNA space, really because we need to have the flexibility and adaptability to make changes as necessary. Our methods need to be robust, and they need to be able to support our clients that move very quickly from perhaps early phase into later phases. And so our methods have to evolve with our clients as well. And so Aldevron has made quite large investments in the analytical method development space to come up with innovative technologies to continue to move the analytical arm of the company in line with the manufacturing. And Aldevron has also put quite a bit of focus on analytical method validation and really moving and cementing that group, or my group, under that quality umbrella. And so really what this has allowed us to do is support what you see here from a manufacturing perspective with a robust analytical platform to make methods, develop methods, transfer methods over into method validation, qualify or validate methods, and then implement them into QC to support the release of these products. And so this is sort of what I'm trying to show here. And this will be a bit of a transition into the next few slides. And really Aldevron's general approach, and we'll have an example, of course, in a little bit about how we're trying to develop and validate standard platform assays and assay panels that ensure product quality and reduce cycle times, ultimately resulting in quicker product release. And so really the name of the game for Aldevron is efficiency, is speed, and certainly trying to release high quality product as soon as possible. And so really from the analytical perspective, where we overlap this manufacturing workflow is by developing these analytical platforms. And so making these robust, validated methods that we can leverage as our clients move through the process. And so what I've highlighted here is that we have certainly done this, and we've certainly validated platform methods to support the plasmid DNA, cell banking, the linear template, and of course the drug substance, which we'll highlight today with the use of SoloVPE for concentration. If we take a look at the analytical platforms offered by Aldevron, and specifically looking at the plasmid DNA and the mRNA drug substance, really the point of showing this slide is really to think more about and drive home and reinforce this idea of platform analytical methods. And so the first thing to note is that these assay panels are aligned with industry and with industry in the guidance, the regulatory guidance, and specifically for the mRNA drug substance assay panel, there was a recent revision, the second edition of the USP Analytical Procedures for mRNA Vaccine Quality Draft Guidelines. And so we've revamped our mRNA drug substance assay panel to be more aligned with the recommendations in that particular document. And keeping on the theme of platform analytical methods, validated analytical methods, we oftentimes supplement our panels as our standard offering with these validated methods. And so these validated methods really serve as a foundation for our assay panels. And really what it allows Aldevron to do is look more at these product specific qualifications, partial validations. It really allows for flexibility as it relates to the mRNA drug substance assay panel. And really the best way and the only way that we can do that is by utilizing technologies that allow us to make platform analytical methods. And so again, that will be put into practice, and I'll demonstrate how we tend to do that in the upcoming slides with the SoloVPE. If we switch gears a little bit and talk about validation, what is validation, what is qualification, it might be helpful to sort of understand Aldevron's perspective in our approach. And I think perhaps maybe a good place to start for this particular slide is to even take one step back. And if we look at the entire analytical procedure lifecycle as described in USP 1220, there's really three separate stages, and it's very straightforward. The first stage is that method development, knowledge gathering, developing, coming up with that analytical control strategy. This is where most of the robustness happens, and this is really the first step in the analytical lifecycle procedure. The second step is this qualification or validation piece that we'll talk more about in a second. That really is a snapshot in time. It is a confirmatory exercise, and so it typically is the shortest of the three stages in the analytical procedure lifecycle. And the third stage that comes after qualification or validation is that continuous improvement, lifecycle management, routine monitoring and trend analysis and continuous improvement. And so once the method's been qualified, validated, implemented into the QC release lab, we need to understand how the method performs over time. And if we start to see problems with the method, then we need to reevaluate how, maybe perhaps redeveloping, things of that nature. And so I think I bring that up really just to give you a context, an overall context of where method validation sits in the overall analytical procedure lifecycle. And so if we think specifically more about analytical method validation, what is it? It really is a confirmatory exercise, right? And it's a documented exercise that a given method performs as intended. Analytical method validations may be conducted in full or in part, depending on the needs of the client and or the phase of the project. And so this is really getting at phase appropriateness, which we'll talk about in a minute. If we take a general overview of analytical method validation process and what Aldevron does, this is very much in line with industry. These steps are very consistent across project to project. We're executing under protocol. We're in quality. We have a protocol approval. We execute a study. We confirm that the method is suitable. As part of that study, we write the necessary reports, get the method approvals under the quality umbrella, and then implement that method into QC. When we think about phase appropriateness, the steps typically tend to be the same, but perhaps we are able to shorten some of them. And so really, as we think about the phase appropriateness and really when it comes to method validation and what the expectations are for qualification or validation, it's important to sort of understand where our clients are in their program and what phase they're at. And so really, one thing that's sort of interesting when we have these types of discussions and we talk to different folks in industry, terminology becomes important. And when we use words like validation and qualification, sometimes they don't mean the same things to everyone. And so for the context of this discussion, qualification in general is an industry standard term that can encompass a number of different activities. But really, when we think about it, when Aldevron thinks about it, we refer to qualifications as partial validations of a method, including selection of a subset of validation parameters to demonstrate method suitability for early phase programs. And so really what that is that it is that partial validation, it is that phase appropriateness. And so if we use that definition in the context of what the FDA recommends that we do for phase appropriate qualifications and validations, I hope that it makes a bit more sense in that when we're saying qualified method for early phases, we're really just meaning the method needs to be suitable, fit for purpose, and that's really what we're looking for, right? So we don't need the full method validation process. We really would want to validate a method once we get into phase 3 and looking into a BLA. And so that's the general approach that Aldevron tends to take, and we have taken. It's quite helpful to our clients to set them up for success, not necessarily over-engineer what we need to do from a method validation qualification perspective, but still deliver a high-quality product for our clients. The one thing I will say that perhaps is, it is aligned with this, but in order for us to, really leverage more of these product-specific qualifications, having platform validated methods where appropriate makes a lot of sense, and that is something that we tend to do. And so we are able to do that when we have the expertise, we have the experience manufacturing the product, we have more information, we have a data set where we can set proper validation criteria, and we do that. And so what we'll talk about today is more in line perhaps with a platform validation, with the idea of laying that foundation such that we can allow these more product-specific qualifications, partial qualifications to come through to expand the scope of our platform method. And so again, it's quite interesting, and it's worth noting kind of how qualification compares to validation. And so really it's quite different, right? And I think that the challenge that we have at Aldevron is we have clients that span the range, right, from early phase to commercialization. And so trying to right size their approach for them can be quite tricky sometimes. And so again, that's where these platform validated methods really help, that are quite broad and can accommodate a large subset of our clients. And so if we look at some of the differences, full-scale GMP manufacturing experience, it all comes down to experience at the end of the day and representative material. And so when we don't have a lot of experience manufacturing a particular construct, if we have perhaps a new method where we're not quite sure of how the method will perform, if we may not have the most representative material, validation is just impossible. You can't do it. And so, and the FDA would agree. And so that's where the qualification comes in. It really is that evaluation piece. And so once we have this qualification and we have a qualified method in place, we start data collecting, right? We gather data. We typically send for qualifications, we'll set wider criteria. And again, this is aligned with industry. And it's important to distinguish sort of this suitable or not suitable to test GMP material qualification versus validated method. The answer is a qualified method, of course, is acceptable to release GMP material. And so when we perform qualifications, we are still adhering to ICH. And so it might perhaps be an abbreviated qualification where we're not looking at all of the ICH parameters, for example, but we still are in line with industry when it comes to qualifications. One thing that's not on this particular slide, which is really quite impactful, is the time and money that it takes to do a qualification versus validation. And so when we think about qualifications, they tend to be quicker. The cost tends to be less. That's something we can get done quickly, and we have flexibility to do that. When we think about validations, they're much lengthier. They take a lot more time, and they're very expensive. And so when we are trying to right size this approach, we don't want to necessarily be charging clients for work on the analytical side that they don't necessarily need it. And in fact, really couldn't have and are just not aligned with FDA's expectations for qualification and validation at that phase appropriateness. And so really just to sum up, the question I get a lot is, why not just validate early? And the answer is kind of twofold. One is that you just can't, right, for the things that we've talked about. And the second is we have that limited sample size. We have a limited data set, so it's very difficult to really do that appropriately. And so we can't set proper criteria for qualification. And so really we do leverage this this qualification approach and this phase appropriateness as we can. And so Aldevron does work with this product-specific qualification approach. And this is sort of a new concept, not necessarily to Aldevron, perhaps not to the industry, but it has become quite useful for mRNA specifically. And so if we think about, historically at Aldevron, manufacturing plasmid DNA, it's much more, that type of material is much more amenable to really platform validations. We've been manufacturing plasmid DNA for a long time. Platform validations are much more straightforward in the respect that the plasmid DNA typically tends to be very similar from construct to construct, from client to client, with respect to matrix, with respect to concentration, with respect to the overall size or attributes of the individual construct. And so we're able to leverage that to really get our platform assays to support release of those products validated. And so really we're set up to get our clients all the way through commercial with that approach. The mRNA space is quite different in that we still do have validated methods. We still can make validated methods leveraging our experience with mRNA manufacturing for some time, but they're not quite as broad as we perhaps would like them as they are with DNA. And so we do need to have this product-specific qualification approach, this, again, phase appropriateness way to bring new constructs into the qualified or validated state of our method. And so typically, Aldevron will take this bracketed approach, experimental design approach, to make a platform validated method which is as broad as possible. But we do need to have a way to bring new constructs into the qualified scope of the assay, into the validated scope of the assay, and that's through the product-specific qualification approach. So really, PSQs at the end of the day explore matrix effects. And so the big difference, perhaps, between a PSQ approach versus a validation, for example, is we're really focusing on the product-specific parameters of the method. So typically that would be specificity, accuracy, and we tend to collect repeatability data because we have it. We really aren't focused on method-specific parameters that we anticipate to not be influenced by the product. So for example, linearity, range, LOQ, intermediate precision, things like that, we leverage our platform validations for, and we really don't expect those to change. And so it really, at the end of the day, PSQs are those partial validations, and they're really only successful when we have robust platform analytical methods. And so what does this look like in practice, this PSQ approach, and how and really how does it relate to serving our clients at the end of the day? And so if we walk through what this PSQ workflow looks like in practice as it relates to mRNA assays, for example, we can use SoloVPE concentration for RNA drug substance as an example. The first assessment really is, do we have a platform analytical method in place? Yes or no? If the answer is no, then straight away we're into, okay, we need to develop, qualify, validate that method. And of course, that takes time. That does take, that is quite expensive. And so in this case, and for our mRNA drug substance panel, oftentimes the answer is yes. And so we can leverage that platform analytical method and move now into this more PSQ workflow, this more streamlined PSQ workflow. And so the next assessment is, okay, the construct that we're looking at, is it within the scope of our validated method? Oftentimes the answer is yes. And so in that case, we feel quite comfortable, right? This phase appropriateness from the analytical perspective, the method is suitable, it's fit for purpose. We can go ahead, no additional work is necessary. That is more or less true. mRNA is, again, unique in that even with an mRNA construct that happens to be squarely within the matrix, the validated scope for a method with respect to matrix and concentration, a lot of these assays are really dependent on the construct and also the manufacturing and process. So double-stranded RNA is an example where most constructs go through this PSQ approach, even if they're well within the validated scope of our method, just because there's uncertainty around what contribution the manufacturing process would have on the presence of residual double-stranded RNA, and also what is the construct's contribution to that as well. But not always, right? And so oftentimes with these platform approaches, we're set up to just test. And so we don't have to do any additional extra work. And that means it's not expensive, and it's very quick for our clients to move through our, to get their products. If we've determined that the matrix or the molecule is not within scope of our validated method, we would move to this evaluation of the matrix, this PSQ approach. And so really the takeaway of this PSQ is to fail quickly. And what that means is we want to understand as soon as possible if our platform method is suitable for the product. If it is, great, we can move forward. If not, we work quite collaboratively with our analytical method development team. And so then that project would move over into analytical method development, and we'd go from there. Sometimes it's a optimization, which could be pretty straightforward, but it also could mean we need to redevelop a method. We need to make a new method. And so again, we want to just understand that quickly. We want to be able to communicate that to our clients as soon as possible, and then set them up for what the next step looks like. And again, if we do need to move into that reinventing the wheel every time when it comes to method validation, it's costly and it takes a long time. And so if we do this PSQ approach, we see no matrix effects, we're really good to go, right? We sort of qualified the matrix, the method is suitable, the scope is expanded. Aldevron utilizes two approaches to do PSQ work. One is an SOP, and so that are more straightforward platform SOP-based PSQ procedures. And so those are the most straightforward. They're very quick. It's a one-day exercise. We test a sample, we understand matrix effects, and we move forward. The other one is a bit more complicated, but really the difference is we have to write a protocol each time. Sometimes we do this if for more complicated methods, but really at the end of the day, it's again very quick. It's not overly expensive when it relates to developing a new method. So again, the idea with these platform methods, right, is we have to have them, we have to be robust, and then we leverage the PSQ process to keep our clients moving forward in their process and bring new methods, new products into scope of the method. Okay, so this will be a point where we can transition into what does this look like in practice, right? And so I've spent a lot of time talking about method validation, phase appropriateness, platform analytical methods. How does Aldevron utilize those? How are those useful for not only Aldevron, but our clients? But now I think it's time to see perhaps what it looks like in action. And so the next few slides will really just highlight a lot of the data that's in the associated white paper for this webinar, and really talk about and demonstrate how we go about validating these platform methods. One thing I will say and reiterate again is that it's platform analytical methods become quite useful when you have technologies that are amenable to a platform approach. And so in this case, it made a lot of sense to utilize slope spectroscopy for this RNA concentration assay because it is so versatile. And so if we spend a minute talking about the study design for this particular validation, Aldevron very closely follows ICH2Q when it comes to validation parameters. This particular assay would fall under the content assay as described in ICH-Q2. And so the following parameters were assessed: accuracy, repeatability, linearity, intermediate precision, specificity, and range. We did perform linearity by diluting the sample to generate levels, 5 levels in this case, which is in compliance with ICH. That's ICH-Q2's recommendations for content assays to create your levels for linearity. Accuracy and repeatability was performed by a 5x3 matrix, which does exceed ICH-Q2 recommendations, which is a typically 3x3 matrix. One of the aspects of this particular qualification was, or validation, was we built in sort of this bridge to Aldevron's existing cuvette-based UV concentration assay. And so really we demonstrated coming out of this study with these data that the methods are very comparable to Joe's point, right? At the end of the day, it's A260 measurements. The readout is the same. And so that's helpful for us as the company, is because when we try to change our clients or switch our clients away from an existing platform to a new platform, even if it's a very sort of similar assay, it does require that extra bridging to give them the confidence that yes, methods are comparable. We won't expect anything to be different from moving to the new method with upcoming manufacturer. In this particular study, we again tried to take the bracketed approach as we've talked about, and so we used representative sample matrices. So we tried to use 3 mRNA sample matrices that are quite common to Aldevron, and so really give the breadth of the study, really allow us to expand the scope. And we did take the same approach with concentration. So we tried to get sort of worst case when it comes to concentration, so roughly 4 mg per mil, all the way down to 0.25 mg per mil. So again, expanding the range broadly, not just with sample matrix, but also concentration as well. And so the next 3 data slides will look very similar. And so I'll perhaps orient you a little bit to what you're seeing here. And so if we look at the table on the left, this will be our matrices that we looked at, TE, uv-vis, sodium citrate. The levels 1 through 5, concentration, theoretical concentration, and measured concentration are reported there as we move from left to right. I am, for the sake of sort of consistency and conciseness, showing just the mean of 3 reportable results for the measured result. And so again, that 3x3 design, 3 analytical runs, just demonstrating the mean here, just to make it a bit more user-friendly to see. So we'll have the theoretical and the measured concentration on those columns for each level, and then starting to look at the validation parameters, repeatability, and relative accuracy. So repeatability is coming from those 3 separate reportable results, 3 analytical runs for each level completed by 1 analyst. And relative accuracy here is reported as the mean relative accuracy. So the mean of 3 recoveries from each of the 3 reportable results for analyst 1. And so what you can see if you just sort of look over this and really looking at the TE result is that the method is repeatable, the method is accurate. So we're very comfortably within our repeatability criteria, RSD, and also our relative accuracy criteria. And so if we look now at more of the intermediate precision component on the table to the right, you can see now we've varied analysts, we've varied day. If we just take a snapshot at level 1 for the TE sample, you can see the 6 reportable result across analysts and day. And the RSD again is really good, well within our acceptable level of precision. And then table 11 shows the linearity results. And so again, as Joe alluded to, method is very linear, and we've demonstrated that here under as part of the validation. If we look specifically at our second matrix, water for injection, a very similar story. RSDs are very good. Relative accuracy recoveries are again well within our validation criteria of 85 to 115%. And if we look at the intermediate precision for level 3 of this particular sample matrix, you can see we've varied day, analyst, and instrument in this case. And again, RSD it's just very nice, right? It's very repeatable, very precise. It really demonstrates, I think, the robustness of the technology. It's very straightforward to use. If we look at table 12, the linearity, again, linearity is comfortably passing our criteria. And finally, if we look at citrate, we can see a similar story emerging. In this case, we're just looking at the lower level, this particular sample matrix where, again, RSD, very good. Relative accuracy across this particular matrix at all 5 levels, very, very good as well. And intermediate precision is the same story. RSD is 2%, well within our plus or minus 10% criteria. Linearity, of course, is, again, very comfortably within our criteria. So really, these data demonstrate at the end of the day, the capabilities of the technology as that platform analytical method. And so just to sum up the slide, the data slides and the white paper, the data presented here really do demonstrate that the SoloVPE variable Pathlength Technology's versatility allows for the validation of a platform analytical method for mRNA content to support our drug substance assay panel. Again, it really does highlight this, this ability for us to demonstrate platform and validate platform methods is so helpful for us. It's so nice because it really does set the foundation for that PSQ workflow. There's several reasons why we decided to move and implement SoloVPE, and really try to summarize them here. And a lot of these things will be reiterating what Joe had talked about earlier in this discussion. The first is this time savings. The SoloVPE is platformable. It allows for validation of platform analytical methods, really resulting in quicker product-specific analytical work, and at the end of the day, quicker product release. The SoloVPE is accurate and repeatable. We demonstrated that across multiple sample matrices, across a broad concentration range. And really that comes from the technology, right? SoloVPE measures multiple data points from a test sample at several path lengths to determine the concentration. The reduction in sample handling, again, to echo what Joe said earlier, is huge for Aldevron, right? It really allows us to take the sample, put it on test, get results straight away. It really does save a lot of time, and also at the end of the day makes a much more predictable method that will support high quality product and really at the end of the day patient safety. The easy to use and robust technology aspect of SoloVPE is instrumental, really. If you have complicated methods, you can develop and validate complicated methods with very difficult to understand analysis. It doesn't always transition and translate well into the QC release lab. And so SoloVPE is an example of something that's very easy to implement into QC release because it's so user-friendly, it's so robust. Some of the data I didn't talk about today that is included in the white paper is several robustness type experiments that we did really to understand how this technology would perform in QC. And it really is remarkable to see how easy this was to implement this technology into the QC release lab. And again, just to hit on the compliance part, in a GMP setting like we all work in, compliance is important, right? And so having the compliance software, having everything we need from an IQ, OQ, PQ standpoint, working with Repligen to be that one-stop shop to provide us everything we need from a GMP readiness perspective is instrumental. The mRNA space requires speed, flexibility, and adaptability. Aldevron is committed to continuing partnerships with leaders in the field, developing cutting-edge technology, as we've talked about here. And so such that Aldevron's analytical capabilities will be a differentiator of the mRNA market. And so again, it is really needing that flexibility and speed to really succeed and to serve our clients, specifically in the RNA space. And I think this is a useful webinar to sort of highlight the work that we do here and this collaborative work that we do with leaders in the field, right? With at the end of the day, keeping the eye on the prize of continuing to provide the highest quality material to our clients. And so with that, I thank everyone for your time. Thank you for listening. I'm happy to entertain.
Fantastic. Thank you very much indeed, Joe and Travis, for an excellent presentation. Without further ado, we'll welcome John and go ahead with some questions. So first question is for you, Travis. Would you be able to elaborate on how you define a platform validation? What does that mean as it pertains to Aldevron, and what happens after the platform validation? How do you implement it across the company?
Of course. And so what I would say is typically Aldevron likes to take that phase appropriate approach for validations and alignment with regulatory requirements. However, it does make sense for us often to fully validate a method prior to phase one. And so the SoloVPE, this white paper is an example of that. Really what it is strict adherence to ICH. We validate the method with the necessary parameters described in ICH 2Q, depending upon the assay type. And really what that is we try to make it very broad, as I tried to explain in the webinar, where we want to make it as the scope as broad as possible. And really at the end of the day, what it does is sets up that PSQ platform. And so it's quick, right, to move clients through, get constructs within the qualified scope of the method. So that's the idea, is to lay that foundation for PSQ. One thing that I didn't necessarily hit on in the webinar that's quite helpful for across Aldevron, not just the analytical folks, so manufacturing, for example, is it allows us to move quickly through engineering into GMP for manufacturing. So if we have these methods in place, we can quickly assess the successfulness of the engineering run based on our platform validations. We can report back to our manufacturing folks, and they're able to make necessary adjustments as necessary and get ready for the GMP manufacturer. So it's really just building that foundation to streamline any further analytical work that's required.
Wonderful. Thank you very much, Travis. We now have a question from a member of our audience, Sarah. How did you go about choosing the extinction coefficient? And I'll just put that out to the panel at large.
So I can start. We leverage just the standard extinction coefficient that's for single-stranded RNA. That was the recommendation on the USP draft guidance revision or edition 1. And so that was our approach. We took the standard approach. That doesn't mean that we have to continue to do that. And so I think, as we talk about this a lot internally, as these methods evolve, as the testing panel evolves for RNA, I think the writing's on the wall, and that we'll have to determine unique extinction coefficients for each molecule. And we certainly can do that. So I think the platform validation approaches and the technology is quite amenable to almost plug and play the different extinction coefficients.
Fantastic. Do you want to add something there, John?
Yeah, again, to what Travis said is it's the space is evolving rapidly, right? In the RNA space. So we do have that flexibility. Right now, there hasn't been a lot of pushback on using those standard extinction coefficients, but we are thinking about that. We have worked with some clients, even doing hydrolysis of the molecule to break it down to its individual components to address that secondary structure type of interferences. So it is something we think about. But again, from the platform or the matrix validation approach, we're trying to cover 90-95% of client needs there, with the notion that there are some larger and more complex RNA structures that where we may get some interference, or where the extinction coefficient may need to be adjusted.
Fantastic. Thank you very much, John. Quick question, another question from a member of our audience, Elliot. Why is linearity required for method validation when the mechanism of the SoloVPE creates a linearity measurement for each sample reading? Isn't linearity already baked into the SoloVPE?
So I could take a crack at that one. So when we talk about linearity, it's really linearity dealing with the reportable results. So just because a 4 mg per mil sample is perfectly linear in the instrument, and a 0.25% sample is perfectly linear, that doesn't mean that their relationship to each other would be linear. So we, in addition to having that linearity built into the system, which we love, by the way, and it helps with troubleshooting and sorts of bridging, you can look at the raw data and get some added assurance that the method is performing as expected. But still, to have this platform validation approach where we're covering a large range of sample concentrations, we still do have to demonstrate that the values that we're getting, the reportable results, are linear with relation to each other.
Fantastic. Thank you, John. Travis or Joe, do you have anything you'd like to add there?
No, well said, John, from your part. And I mean, agreed. The fundamental basis of the technology is proving that linear relationship for absorbance over path length, taking multiple reads. So it's critical from our side of it that our pass-fail criteria is quite aggressive, being usually 3 nines or higher, to be deemed as a valid value. And John's answer certainly covers the other part of why you would do that.
Fantastic. Thank you very much, Joe. Okay, another question from a member of our audience, Lily. How were the theoretical values of concentration determined for accuracy in this validation? Was a certified standard used, or did you determine concentration with the traditional cuvette method prior to validation?
We used the traditional, our platform method to establish the theoretical value.
Excellent. Thank you very much.
Okay. Yeah, and just quickly, as Travis pointed out earlier, that did allow us to incorporate the bridging by using that as the theoretical value from the cuvette method and going with the and comparing that from an accuracy standpoint. So we killed two birds with one stone with that.
Fantastic. Thank you, John. Okay. And another audience question. We've just got time for perhaps one more. So a question from Jessica. From sample to measurement, how long did it take? Is there a possibility to use this with the VPX?
I can certainly take that one. So data acquisition occurs in less than 1 minute. So the only responsibility the analyst has after before they test the sample is just entering the method information into the software and pipetting your sample into the vessel. So about all in all, you're looking at a little less than 2 minutes of measurement, and that would include the sample prep. The answer to your question is this possible with the VPX? And the answer is yes.
Great stuff. Thank you, Joe. I think we can squeeze one more in very quickly. One for you, actually, Joe. How long does it usually take for the SoloVPE system to be installed and validated?
Sure. So selfishly, we won't be the bottleneck in that operation. So usually the turnaround from order to ship to IQ, OQ occurs, I want to say, anywhere from, say, 4 to 6 weeks on average. The real benefit of our technology, again, is working with customers that have the project demand to implement the technology. So the higher the project, the closer our applications team and support teams get to work with the customers to expedite the process using a variety of different tools that we can offer to speed things up. So on average, I'd say this is a 6 to 12 month operation, provided that you have the resources and the logistics to give it the proper attention it needs.
Fantastic. Thank you very much, Joe. Well, unfortunately, that's all we've got time for. Many thanks to Joe, Travis, and John for answering those questions and for the presentations. That's unfortunately it. But any questions we didn't get to today, rest assured, will be responded to by email. This webinar will be available on demand tomorrow. Look out for an email from us with the link. And all that's left for me to do is to thank once again Joe, Travis, and John for their participation today. And thanks to our audience for listening in. We hope to see you again soon. Bye for now.
Thank you, everybody.
Thank you.